So we're going to go into now population genetics, which is also tested quite a bit. And the main thing to mention is the Hardy-Weinberg equilibrium. So population genetics is a field of biology that studies the genetic composition of biological populations and also the changes in genetic composition that result from the operation of various factors, which also includes natural selection. But for the Hardy-Weinberg, so that was the general field of population genetics. But for Hardy-Weinberg equilibrium, it really focuses on that, like, genetic variation will remain constant. So here I say here a principle stating that the genetic variation in a population will remain constant from one generation to the next in the absence of evolutionary influences. And we'll go into the next slide, like what are the requirements in order for us to be able to use these Hardy-Weinberg equilibrium principles. So one thing to know before going into the equations is what allele frequency is. And allele frequency is the proportion of the allele in total alleles of a population. So if we're using the big A and little a example again, so if we're looking for the proportion of that big A allele, then it would be that big A allele. So let's just make it simple. We have two organisms. One organism is homozygous for the dominant allele, so big A, and then the other organism is heterozygous, so has big A, little a. So we're trying to find the proportion of that big A allele, then we would be able to know that homozygote for the dominant one would be that allele times 2. So you would have 2 A's from that homozygote plus the 1 allele from that heterozygote. So you would have 3 of the big A's over like the total alleles of the population, in which case, since you have 2 individuals, times 2 for 2 alleles per person, and then you would have 3 out of 4. So in that particular group of 2 individuals, the proportion would just be 3 out of 4 of the big A. And the main result of that Hardy-Weinberg equilibrium are these Hardy-Weinberg equations to calculate for 2 types of alleles in a population. And so usually the P plus Q equals 1 is usually P is considered the dominant allele and Q is the recessive allele. And this particular equation is usually related to the allele frequency. So like we mentioned a little earlier. While this other equation, which is derived from the original one, is P squared plus 2PQ plus Q squared equals 1, which is a, it would be like the genotype frequency. And also both of these equal 1 because, so for frequencies, right, we would have that in the previous example, 3/4. And then the other proportion of the alleles would be 1/4. So, you know, all of these frequencies would be less than 1, but they all should add up to that 1 or 100%. And these are just conditions that need to be true for Hardy-Weinberg equilibrium to occur and for us to be able to use those equations. So one is that, you know, we have to have a large population. And the reason is for mathematical calculations, it's used to assume that the population is infinite for those calculations. Also that there are no new mutations. So everything is kind of as it is already as it started, nothing new is coming in. And going on that path, no migration of organisms into and out of that population. And so no changing from there. However, it is required for there to be random mating. So in the case that genotype doesn't have an influence on an individual's choice of mate. So there needs to be random mating in order for this, as in they can't only want to mate with a certain group, because then you would have a disproportionate amount of individuals. And then also no selection. So genotype, in which case genotype has no influence on fitness, which in fitness, in evolution and natural selection, you guys might already know, is the ability to reproduce and transmit genes to the next generation. And so I have another question here. So sharing the results here, we have 33% saying A, we have 33% saying D, and we have 33% saying I have no idea. Okay, so you can understand this is a little bit complicated. Just hopefully you guys are reviewing this when you are, studying for the biology portion. But here, I've kind of laid out a little bit of the math of what we're supposed to try to do. So let me so the answer is A, 0.64. And let me just go back a slide so that this math is seen. But here, the question says, there is a ladybug population in which 16% of the ladybugs has a homozygous dominant genotype. Red is dominant to black. So that means that this dominant is, so this AA would be, red, because this is the dominant denotion. Usually for like recessive, it's always lowercase. So here, dominant, red is dominant to black. How 0. Okay, see, I will definitely get to this. So assuming complete dominance and Hardy-Weinberg equilibrium criteria met, what is the frequency of ladybugs that display the red phenotype? Right, so we know that red is that dominant phenotype. And so here we have 16% have the AA genotype. And so here we know that 16% have to be, this AA genotype. And going back to the, I'm looking for, yes, the pen. So going back to the second Hardy-Weinberg equilibrium equation that was related to genotype frequency, this p squared is referred to, is associated with that AA frequency. This 2pq is related to that heterozygote frequency, and the q squared is related to the homozygous recessive frequency. Right, so this is just something to know. So then we know that this p squared value is the AA. So here we then know that because 0.16 is AA, so this p squared value is 0.16. In order to find the, the Q based on the first Hardy-Weinberg equation for allele frequency, we need to find this p value. This value for p, which is why then you would square root that 0.16 to get 0.4. And then this is why we would do the 1 minus 0.4 to get the Q value. And the reason why I got the Q value is because, right, we are looking for the frequency of ladybugs that display the red phenotype. So here we know that the red is dominant to black, which means that red, actually, don't look at that part anymore, but here, red would be any of these genotypes, right? Because that dominant phenotype is going to be shown as long as you have one copy of that dominant allele. However, we know that if we look for the black ladybugs, we can just subtract that from the original one, this one. If we just subtract this one minus the the genotype frequency of this recessive, then we can find all of the ones that have this, dominant red phenotype. So this is why I decided to look for Q instead. And so for Q, we know it's 0.6. And then when you square Q, you get 0.36, right? But this is the frequency of just that. So in order to find the reds, we would have to do 1 minus 0.36. And then that's how you would get the 0.64. So hopefully, does that make sense to everyone? Okay, great. Yeah. So these Hardy-Weinberg, problems are a little bit, they do take a little bit more time, but they usually always give you like a value that's easily, um, it's easy to find the square root of. So like that 0.16, 16%, 9%, 25%, these easily square rooted numbers. Also, just a side note is that you could also, find the individual frequencies for each of these and, add it up and you would also get 0.64. It's just that I feel like this other way is a little bit faster in terms of if you're running short of time or just, you have that idea to do that instead, because that is also another solution.